Okay, so let's dive back into it here. Now, this figure sort of shows us with some colors and some lines and so forth, sure, a little bit more about what was there. We don't need to worry for the moment about no cooperativity hypothetical, because I'm going to explain what cooperativity is in a minute. But you can see from this graph the difference, the real significant difference between the oxygen concentrations. Here's the concentrations in the lungs. We see that it's high enough that hemoglobin is loaded with oxygen, just like myoglobin is. But when we get out to the tissues are really needing the stuff. And you can see hemoglobin has given up a lot of oxygen compared to what myoglobin has given up. At this concentration, myoglobin has only given up 7%, where hemoglobin has given up almost 2/3 of its oxygen. So hemoglobin is important for us to be able to pick up oxygen where it's available and drop it off where it's needed, a very, very important property that hemoglobin has. So I promised I would talk about cooperativity. So to talk about cooperativity, I need to go back to look at this little tiny movement of the iron. Now, as I said, this happens in myoglobin and hemoglobin both, but I'm going to distinguish them for you in just a second. For the moment, we're focusing on the heme group, and we can be thinking about either myoglobin or hemoglobin. Oxygen binds, and what you've seen is that that iron atom moves about 0.4 angstroms. Well, that isn't independent of everything else. In fact, that iron atom is attached to a histidine in the protein. Yes, another histidine. And you'll see there's a lot of histidines to think about in hemoglobin and myoglobin both. So the iron atom is attached to the histidine. If I pull up the iron, I'm going to pull up the histidine. Right? Well, if I pull on the toe, and the toe bone's connected to the foot bone, and the foot bone's connected to the ankle bone, and the ankle bone's connected to the shin bone, you guys begin to get the idea. I'm not going to sing, but you begin to get the idea that making a change here may have changes that now resonate all the way through the rest of the amino acid sequence. In fact, that's exactly what happens. This tiny change right here will change the shape of that protein ever so slightly. You say, well, so what? So it changes the shape of myoglobin. What happens? And the answer is nothing. You'll see why changing the shape in hemoglobin, however, is important. When you change the shape of one of the subunits of hemoglobin, the subunits of hemoglobin are interacting with the other subunits of hemoglobin. So now this change that you saw in the one subunit is communicated to the other subunits. The effect that has is it changes the affinity that those other subunits have for oxygen. They want to bind oxygen more readily than the first one did. They bind it much more easily. So that means that the second oxygen will bind more fast, more readily, because of that very subtle change that happened in the first one. Well, the second one now induces the third one to change, and the third one induces the fourth one to change. So what we've seen, and we look at this plot that was here, when we look at the plot of the oxygen binding of hemoglobin, is we saw at low concentrations, it didn't bind very much, but once we got one binding, boink, and the second one binds, and we get a boink, we're changing the way that hemoglobin binds oxygen depending upon how much oxygen actually is present. That's really useful if we need to have flexibility in our binding and in the release of oxygen. So I've just defined for you, not formally, but I've defined for you practically what cooperativity is. And now I'm going to tell you what it is in a formal sense. Cooperativity is a phenomenon where the binding of one molecule to a protein affects the binding of subsequent molecules to the protein. Now, we see that the binding of the first one favors the binding of the second, favors the binding of the third, favors the binding of the fourth, and we see now why those subunits really made a difference. Myoglobin doesn't have anything else to interact with. It's a lone ranger. It's all by itself. It can change its shape, but so what? It's like if the tree falls in the forest and there's no one there to hear it. Is there any noise? Okay, same problem. The same thing can happen when hemoglobin gets out here to the tissues and it starts to lose its oxygen. Remember, these aren't covalent bonds. These are attractions. It loses one oxygen, and now what's going to happen? It's going to favor the loss of a second. It's going to favor the loss of a third. It's going to favor the loss of a fourth. That's really good because as hemoglobin travels further in the bloodstream, more and more the oxygen concentration is decreasing. The need for oxygen is going up. Hemoglobin is really, really good at supplying oxygen where it's needed. Does that make sense? Now, hemoglobin's got more built into it than just that. Okay? It's got more built into it than just that, and I'm going to show you some of that. So we've seen now, okay, the basic structure of hemoglobin is such that it can, in fact, provide varying amounts of oxygen depending upon needs. All right? Talk about your subtle changes. Look at that. Okay? Deoxyhemoglobin versus oxyhemoglobin. At this point, I need to introduce another term to you. All right? The guy on the left was deoxyhemoglobin. And we saw on that plot that it took a certain amount of oxygen concentration before the hemoglobin woke up. Why did it take a certain amount of oxygen before it woke up? Well, we hadn't converted any of the subunits yet. All of the subunits were in a form that didn't want to bind oxygen nearly as much as they were once the hemoglobin started binding oxygen. We give these two different states names. Okay? A low affinity binding state is something we refer to as the tight state. And I like to think of it as an uptight person. Okay? An uptight person isn't very flexible. They don't adapt very well. Right? They're very rigid. You know people like this, I'm sure. Okay? And that T, we call it the T state, that T state doesn't bind oxygen very well. It can. If it didn't, we wouldn't bind any oxygen at all, but it doesn't bind very well. We have to get some more oxygen in there before it really takes off. Once it flips, it's more relaxed, oxygen has greater access, and that relaxed state is called the R state. So the T state is low oxygen affinity. The R state is high oxygen affinity. Hemoglobin is flexible. One of the properties I said about proteins that was important for their function was their flexibility, and it's their flexibility that allows it to change from a tight to a relaxed state. And though that looks like a very subtle change, it is. Right? I told you that with that 0.4 angstrom movement caused these things to happen. Hemoglobin is changing. Now, I said that 0.4 angstrom movement is responsible for us being animals. We have to have this varying oxygen carrying capacity because we have varying needs for oxygen. If I'm late to class and I'm rushing to get over here, my oxygen need goes up. If I'm running away from a grizzly bear that's chasing me, my oxygen need goes up. If I'm sleeping, my oxygen need goes down. Animals have widely varying needs. Plants don't have that. Bacteria don't have that. We have that. Okay? We have to have this movement. All right. Now, here's the T to R. Okay? So, and that's graphically, actually we won't worry about that. Don't worry about that. All right. All right. Now, this is trying to show you what I told you in words before. We've had oxygen bound, not bound down here, and then they've tried to show on the same figure oxygen bound, and you see a different green blob, and you can actually see it's moved up. And that's showing here's the histidine pulled up when oxygen bound. Here's the same histidine not pulled up when it's there. It's just, again, showing you, trying to show you in one figure what I said in words earlier. Okay. Now, we now have the tools we need to understand some other really amazing things about hemoglobin. The first of these is a very interesting compound called 2,3-BPG. We'll talk about it later in the term. 2,3-bisphosphoglycerate, and yes, you can call it 2,3-BPG, is a molecule that is produced by rapidly metabolizing cells. Well, what's a rapidly metabolizing cells? Well, if I'm running away from that grizzly bear, my muscle cells are rapidly metabolizing. They're burning glucose so they can get energy to contract and get me out of there. And to get that energy from glucose, they need oxygen, okay? Because there's all kinds of reactions going on. One of the byproducts of their metabolism is this molecule right here. This is a very, very important metabolite. It's a very important byproduct. Okay? Why is it important? Well, it's important relative to hemoglobin because when we look for our cursor, there we go. Okay? When we look at the structure of hemoglobin, okay, what we discover is nope, I got the wrong one. Where was I? No, that's not it. Well, come on now. I'm going to lose my point just because of what I'm trying to find here for you. Here we go. No, that's not it either. Blast it. Stupid professor. I've only given this lecture about 300 times. Here we go. All right. Now, subtle change here. When I started the lecture and I showed you a structure for hemoglobin, I said, think of that donut hole. The donut hole is important. The donut hole turns out to be a perfect place for 2,3-BPG to bind. A perfect place. It has the exact molecular dimensions of 2,3-BPG. And yes, it's going to fit in this little tiny hole right in the middle of hemoglobin. Okay? What does 2,3-BPG do when it binds to hemoglobin? It stabilizes the T state. Now, based on what I told you about the T state, what happens to the oxygen affinity of hemoglobin for oxygen? What's the affinity of hemoglobin for oxygen in the T state? It goes down. So 2,3-BPG is waving to the hemoglobin saying, hey, I'm a rapidly metabolizing cell because I'm a byproduct of this. Oh, there's hemoglobin. I'm going to grab onto a hemoglobin, and hemoglobin's going to say, oh, and it's going to let go of oxygen. So 2,3-BPG has just served as a flag for a place to drop oxygen. So in addition to the cooperativity that I talked about before, here is another specific mechanism that can be used to give oxygen to cells that need it the most because they're rapidly metabolizing. Rapidly metabolizing cells need the most oxygen. And you're thinking, well, okay, that's great, but then how in the heck does it get out of there? Well, keep in mind that like oxygen, 2,3-BPG doesn't bind covalently. So it can come off, it can go on, it can come off, it can go on, right? When it leaves that rapidly metabolizing tissue, it comes off, it goes on, it comes off, it goes on. And sometimes when it comes off, it gets grabbed by one of the nearby cells because it's actually a source of energy. The cell grabs it, and hemoglobin makes it back to the lungs without the 2,3-BPG. If it gets back to the lungs with the 2,3-BPG, what's going to happen to the ability of hemoglobin to carry oxygen? Down. It's still going to be in the T state, right? Now, guess what? Smokers have more 2,3-BPG in their blood. One of the reasons that smokers get out of breath with even light exercise is because they have a much higher percentage of their hemoglobin that makes it back to the lungs in the T state, stuck to 2,3-BPG. Okay? So there's a very real, very physiological phenomenon that happens based on this individual molecule. Anesia? So is that because when you're a heavy smoker, your body metabolizes things faster? Let me save the answer to that question when I talk later in the term about glucose metabolism. The answer is basically yes, but you don't know why yet. Okay? But there's a very important reason why that's the case. It's actually a catch-22 that smoking causes for this phenomenon to happen. Okay? Part of the problem is the fact that smoking also produces carbon monoxide. And carbon monoxide, one of the problems with carbon monoxide is it binds in the same place on hemoglobin that oxygen does. It competes with oxygen, so if you have carbon monoxide that you're getting regularly from smoking, you're also reducing your oxygen carrying capacity. Now you've got a really reduced load of oxygen that's being delivered. When cells run out of oxygen, they have to metabolize faster to make up the difference. So there's the short answer to your question. Okay? Okay. So 2,3-BPG, really fascinating, really important molecule that we need to understand. We'll talk later in the term about why and when it's produced. It's actually produced as a byproduct of the breakdown of glucose. Okay. Here's the effect of 2,3-BPG. All right. We see that 2,3-BPG is, in fact, lowering the oxygen carrying capacity. It's favoring the release of oxygen, which is great. Okay? But if it gets back to the lungs and it's still there, we're going to have more of a problem. Okay? So we don't want to have 2,3-BPG. You can see way down here, the same oxygen concentration. We're not getting as much hemoglobin bound with oxygen. And as a consequence, we are not going to have the same oxygen carrying capacity. Okay. That's actually showing it kind of cool in the middle there. But the molecular dimensions between there is exactly that for binding 2,3-BPG. You can see histidines, you can see lysines. Histidines are all over in hemoglobin. Histidines are very, very important constituents of hemoglobin, as we shall see. Okay, now, 2,3-BPG is important for another reason. Okay? Let's think about fetal hemoglobin. Fetal hemoglobin is an important consideration because fetuses need oxygen, but they're buried deep inside of mom. And mom has a certain oxygen carrying capacity. However, if mom's hemoglobin is fighting with the fetus for the same oxygen, the fetus is going to come out on the short end. All right? Well, it turns out the fetus has to have a built-in advantage, and the fetus does have a built-in advantage. Okay? The fetus has to be able to take oxygen away from mom. Okay? That means it needs to have a form of hemoglobin that has greater affinity for oxygen than mom's hemoglobin does. And it turns out it has that. Fetal hemoglobin is slightly different from adult hemoglobin. What I've shown you so far is adult hemoglobin. Fetal hemoglobin replaces the beta subunits with gamma subunits. Again, they're very similar, but they're not identical. So fetal hemoglobin has 2 alpha and 2 gamma subunits instead of 2 alpha and 2 beta subunits. Okay? Now, the gammas are different enough that that little donut hole in the middle won't accommodate 2,3-BPG. As a consequence, fetal hemoglobin remains in the R state. It doesn't flip back to the T state very easily at all. If you recall, the R state was the higher affinity state where it bound more oxygen. It's because of this tendency for fetal hemoglobin to be in the R state more than mom's hemoglobin is that the fetal hemoglobin can take away oxygen from mom's hemoglobin. That's exactly what we see here. Okay? Greater affinity, it's flowing in the direction of the fetus, and the fetus is safe. And you're sitting here thinking, but wait, the T in the R state made sense because it gave oxygen according to my varying oxygen needs. Isn't the fetus going to be in trouble? Doesn't it have to give up that oxygen? The answer is it does give up its oxygen, just not as readily. The needs of the fetus for oxygen are not like mom's. They're much more constant. The fetus is not running up and down stairs. The fetus is not jogging. The fetus might be kicking mom, but that's about all that the fetus does. Okay, so the needs of a fetus for oxygen are fairly constant, and so the fetal hemoglobin doesn't have to be as variable as mom's hemoglobin has to be. And it works out very, very well. Shortly after birth, within about the first year after birth, fetal hemoglobin ceases to be synthesized, and and adult hemoglobin starts to be synthesized. And so that's a transition that happens. We code, however, for the fetal hemoglobin all of our lives because it's in our DNA. We do make a small amount of it even as an adult, but most of the hemoglobin that we make is adult hemoglobin. Now, I tell you that because there are some diseases that are known where drugs have been designed to stimulate the production of fetal hemoglobin because adult hemoglobin has a problem. And this has been useful in treating some diseases like sickle cell anemia, where sickle cell anemia arises from a defect in the hemoglobin that causes the hemoglobin to form sickle cells. And let me save and explain that in a bit. I'm getting a little ahead of myself. But suffice it to say that if we can stimulate the production of fetal hemoglobin in an adult, we may be able to treat certain hemoglobin-related diseases in adults. Okay. Now, as if that wasn't enough, there's even more about hemoglobin. It doesn't stop. I told you it was cool stuff, right? It doesn't stop. Hemoglobin has yet another mechanism, okay, to make sure that tissues that need oxygen get it. Okay? It is an amazing protein, an absolutely amazing protein. Okay? This other phenomenon I'm going to tell you about is called the Bohr effect. You can see it schematically here. It was discovered by a scientist named Bohr, no surprise. And the original observation was very simple. If you look at the oxygen binding affinity for hemoglobin at pH of 7.4, and you compare it to a pH of 7.2, a very slight acidification, you see that the affinity for oxygen goes down, meaning that hemoglobin is giving up oxygen. It's releasing oxygen as the pH is going down. Now, physiologically, this has enormous consequences. First of all, our pH doesn't change very much in our bloodstream. This would be a pretty good change in our bloodstream. When does pH go down in our bloodstream? Well, it turns out that rapidly metabolizing tissues release protons. So in addition to having 2,3-BPG being released out there, our rapidly metabolizing tissues are making and releasing protons. There's another flag that says, hey, over here. Those protons, well, now we're going to talk about the molecular basis by which this occurs. But the phenomenon is simple. Protons favor the release of oxygen by hemoglobin. At a molecular basis, how does that happen? Okay? Well, within hemoglobin, we have histidine residues. Histidine residues have the ability to gain a proton. And if you recall when I talked about the R groups and the pKa's, I said histidine was an interesting one in that it was able to gain protons at a pH reasonably close to the pH of blood. The pKa was 6, and the pH of blood was about 7 to 7.4. Of all the amino acids, histidine is the most likely one to gain a proton at that point. When histidine gains a proton, what it does is it changes its charge from 0 to +1. When it changes its charge from 0 to +1, not too far away is a negatively charged aspartate. What do you suppose happens to the interaction between the two? It goes up. When they interact, plus attracts negative, what happens? Are we going to see a slight movement? You betcha. Is that slight movement going to change the properties of the hemoglobin? It is. Is it going to favor release of oxygen? It does. And now we've seen how that phenomenon occurs. Okay? There's other things that can happen with that binding of the proton. Okay? Look right here. Here is a group, it could be a histidine, for example, that has a proton on it. Another thing that's being produced by rapidly metabolizing tissues is carbon dioxide. Carbon dioxide is a byproduct of metabolism. Just like 2,3-BPG, just like protons, Carbon dioxide is a byproduct of metabolism. Our body has two ways of carrying it back to our lungs. One is by simply dissolving it as bicarbonate in our blood, and it turns out that's an important consideration for maintaining blood pH. Okay? The other is it can bind to something like this nitrogen with this hydrogen on it and form a carbamate. The carbamate released, look at there, there's another proton. This is yet another way to get proton concentration up because this guy is binding to nitrogens that have hydrogens on them. Okay? Now, what we've done, if this was a histidine that grabbed a hydrogen and now we put a carbon dioxide on it, that hydrogen just got recycled. It can go out and stimulate and bind to another histidine and stimulate this process. We see a snowballing of the process. All right? This is really a very efficient way to get more and more histidines full of carbon dioxide to release more and more protons and thus stimulate release of more and more oxygen. It's a snowball process. Okay. Now, what do I want to say there? There's something else I wanted to say about that. When we get to the lungs, this process will get reversed. Carbon dioxide will get released from here. We go back to our original state with our hemoglobin. And this happens partly in our lungs because oxygen is present in very high concentrations. Oxygen present in very high concentrations causes, it literally forces itself onto hemoglobin. It changes the shape of the hemoglobin, and that change in shape ultimately reverses this reaction to favor the release of carbon dioxide, which we exhale. That's essential for the movement of carbon dioxide in our body. All right, but what else do I want to say here? This shows how we can dissolve carbon dioxide in our body. Okay, carbon dioxide can be solubilized with water to make carbamic acid, or carbonic acid, it's not carbamic, carbonic acid. Carbonic acid readily ionizes to make bicarbonate and protons. This is one of the ways that we're getting protons near rapidly metabolizing tissues. And last, carbon dioxide, because of that binding that it's doing onto those nitrogens, and by the way, for those nitrogens, I'm essentially talking about them as if they're all on histidines. And that's what I'll hold you responsible for. But what this tells us is that here's the Bohr effect. The Bohr effect says not only as pH drops do we see a release of oxygen, but so too do we see a release of oxygen when CO2 binds to hemoglobin, because compare the blue to the purple. The purple has the lowest line. The only difference between the blue and the purple is carbon dioxide has bound compared to the blue. Okay? Wow, a lot of things happen in the body to allow us to carry oxygen, to grab carbon dioxide and move it away. Now, I want to emphasize something here, and that is that carbon dioxide is not, underline not, binding to the heme group. Okay? It's not binding to the heme group. The only things that bind to the heme group are oxygen and carbon monoxide. Carbon dioxide is being carried on those nitrogens of histidines and related side chains. Okay. Now, I want to spend just a few minutes talking about some genetic considerations. I'm actually doing fairly well on my time here. Maybe I might even finish a little bit early today. I mentioned briefly earlier sickle cell anemia. And sickle cell anemia is a disease that affects the globin genes that make up hemoglobin. So you've seen alpha, you've seen beta, and sickle cell anemia can affect, actually it can affect either of those. And there's different genetic mutations that can cause the phenomenon I'm getting ready to describe to you to happen. When certain mutations happen inside of hemoglobin, there is a phenomenon known as I said, sickle cell anemia. And the reason that cells sickle is as follows. The mutations in the globin, whichever globin it is that happens to get that mutation, the mutation that happens in the globin changes an amino acid from, typically from a lysine into one that is nonpolar. That's the most common way, and there's many different mutations, but that's the most common one. When that happens, the hemoglobins inside of the blood cells will polymerize when oxygen concentration is low. Polymerize meaning if I had one single hemoglobin over here of 4 subunits, it will link to another one, to another one. I'll make a chain of these 4 subunit hemoglobins that goes very long. That happens when the oxygen concentration gets very low. Where does oxygen concentration get low? Well, it gets low for exercising. People who have sickle cell anemia will discover that they do even moderate exercise. They get very, very painful feelings in their muscles. And the reason it's happening is because when you're exercising, your blood is giving up most of its oxygen. And as it gives up its oxygen, the hemoglobins are polymerizing, and the polymerizing of the hemoglobins convert the nice round blood cell into this sickle shape. It's actually the polymerizing that does this. One of the reasons it's painful is A, the muscle is starving for oxygen, but more importantly, the capillaries are plugged with these things. There's nothing moving through the capillaries because they're plugged up with sticky sickle cells in contrast to the smooth, rounded blood cells that's present for non-sickle cell disease. So it's a very painful disease. It can be fatal. And it is an interesting disease, but let me come to the interesting part in just a second. I want to say a little bit about why we call it an anemia. The reason we call it an anemia is basically a lack of blood cells. And in sickle cell anemia, what happens is the body, when it encounters a cell that looks like this, thinks it's been damaged and takes it out of action. So a person with sickle cell anemia will constantly be struggling to supply enough blood cells because its half-life of blood cells is very low compared to a person who doesn't have that disease. Our blood cells stay round for, I don't know, a month or 2 months or something like that. In a person with sickle cell anemia, it varies with how much exercise they do. And so their blood count, that is the blood cells per milliliter, can go down quite low if the body is pulling these out of action. So that's why we call it an anemia. Now, one of the questions people have had about sickle cell anemia, as they do about any genetic disease, is, well, why is it so widespread in the human population? Shouldn't it have been selected out? Because most genetic diseases that we see aren't really very widespread. Some are, but most of them aren't. And when we see a disease, a genetic disease that is very widespread, we think that there has to be some sort of advantage that it gives to people who have it. And so epidemiological studies that that have been done on sickle cell anemia turned up an interesting fact. And that is that the greatest prevalence of sickle cell anemia arises in locations in the world where the malaria prevalence is the highest. Sub-Saharan Africa being one of those. Okay? Now, anybody can get the sickle cell anemia is not unique to any ethnic group, but it's more common in Sub-Saharan Africa and other areas of the tropics than it is in non-tropical areas. And when people then took that information and examined the survival of people who had at least one gene for sickle cell anemia, what they discovered was that people who were heterozygous for it, that is, they had one good and one bad gene, they had a greater likelihood of survival of malaria than if they had either 2 good genes or 2 bad genes. The best survival was those who were heterozygous. There was an advantage. The organism that causes malaria is dependent partly upon oxygen that it derives from blood cells. Now, it's not completely understood why heterozygous works. Perhaps there is something different about a low level of production of this defective protein that is actually interfering with the malarial organism. But suffice it to say that the evidence is strong that there's a protection against malaria given to people who have sickle cell anemia. Okay. There's the traits there. And there you can see the percentage of the population. You can see this is the falciparum malaria. There's different types of malaria, by the way, but this overlays very interestingly over the same locations that have exposure to it. Okay. Questions about that? We've gone through a lot of material today. Everybody's going, yo. Should we finish the week with a song? Okay. I have a song that will actually summarize all of this hemoglobin stuff for you, and then we'll call it a week, the first week. Okay, this is a real easy one to sing. It's called Hemoglobin's Moving Around to the tune of Santa Claus is Coming to Town, okay? Oh, isn't it great what proteins can do, especially ones that bind to you what? You can't see it. Oh, man. This stupid screen, it doesn't match up. Okay, let's try it again. Oh, isn't it great what proteins can do, especially ones that bind to O2. Hemoglobin's moving around. Inside of the lungs, it picks up the bait and changes itself from T to R state. Hemoglobin's moving around. The protoporphyrin system, its iron makes such a scene, arising when an O2 binds, pulling up on histidine. The binding occurs cooperatively, thanks to changes quaternary. Hemoglobin's moving around. It exits the lungs, engorged with O2, in search of a working body tissue. Hemoglobin's moving around. The proton concentration is high and has a role. Between the alpha betas, it finds imidazole, that's histidine by the way, to empty their loads. The globins decree, we need to bind to 3-BPG. Hemoglobin's moving around. The stage is thus set for grabbing a few cellular dumps of CO2. Hemoglobin's moving around. And then inside the lungs it discovers oxygen and dumps the CO2 off to start all over again. So, see how this works? You better expect to have to describe the Bohr effect. Hemoglobin's moving around. That's a hint at the end, by the way. So. All righty. See you guys on Monday. Have a good weekend.